Do you have a link to that that you could share?
Yeah, I'll put it in the slide. So, but essentially, this is the thing about art. People are scared to put themselves out there. A lot of people are scared to do science as well because they think they can get it wrong, just like you think you can do art wrong. And it's all just about exploring reality. And so this is my entrance. This is on the right here. You can see my Martian garden. I've been growing it in the basement, and it's been put onto the surface of Mars there. And this is just how I use like horticulture, like the playing with plants, just for aesthetic purposes, as a way to get used to killing plants and used to doing things wrong. So I work out what does right. The true scientific methodology is just keeping notes as you go, right? So you don't make the same mistake too many times. But so many people are worried about making mistakes and worried about putting themselves out there. And sometimes you've just got to do that, to learn and to grow. And so we can use these artistic and gaming journeys to take our minds to alternative places. What we have here is some students playing Settlers of Catan. It's a classic board game. And I was then throwing into them like challenges related to going to Mars and related to climate change on Earth, and watching the kind of gaming mechanisms change and how they interacted. And afterwards, one of the students decided to draw this storyboard about a couple of people being on Earth, thinking about going to the moon, and then thinking about going to Mars. And so they've made themselves this little storyboard in which we can then build the mechanisms and technologies that need to be developed to actually help humanity go on that journey. And so from that one little like throwaway statement that he made all those years ago about growing a rose on Mars, suddenly now we have this whole new set of like science fiction, art, storytelling opportunities all being created at once. This is a Cambrian explosion of imagination opportunities for the internet generation. And let's just hope they have a collective goal which is worth aspiring to. Okay, cool. And so the focus of this talk is actually nod factors. The power.
Thank you, Richard.
Really nice to meet you, Jen.
I would actually love to follow up with all of you later. You're an amazing group, and so please let's share contact info.
Well, you're now part of the group, Jancy.
So aw, thank you so much. Have a great rest of your day. Bye.
Take care.
Hey, Richard, right before you get into the awesome science, can I add to one thing you were saying? It was here on the chat, and it's actually super relevant to this whole conversation. So Elon Musk is trying to go to the moon. He's got this awesome moon rocket, and there is a Japanese billionaire called
Yusaku Maezawa.
Maezawa-san, yeah, Maezawa-san.
Yeah. Cool. She was ready to go too. Are you on that rocket?
This whole thing in Japan, when it got announced, it was such a big deal because one, he's Japanese, and people here, anytime there's a Japanese person prominent in media worldwide, they make a big deal out of it. But the whole concept of him taking a handful of artists from different means on a trip to the moon and back in order to produce art to inspire humanity to pay attention to not only the moon and space sciences, but to the earth that we live in. It's such an amazing concept. It ties into everything we were talking about this evening and what you just talked about now. It's a trip that I wish I could go on. I think all of us wish we could go on, but I can't wait to hear the compositions that are going to come back from that trip, and the paintings that are going to come back, the architecture that's going to come back from this trip. It's going to be fantastic. So if you haven't already, I'm sure everyone knows. Chat group has already done so, but check out Dear Moon. It's a cool concept that I can't wait to see what comes out.
Yeah, I also think it's quite interesting because Dr. Barker actually brought it up a couple of live sessions ago, and it really changes your perspective as well, because everyone's looking at they're only looking at Maezawa, they're only looking at the billionaire himself, but they're not looking at the art aspect of things. And I remember Dr. Barker saying a few sessions ago, when these artists come back, look at how it's going to change the state of everything on Earth, where it's going to create this massive boom around art with regards to that. And how is it going to change the way we look at space, we look at science, and we look at art itself? It's something that should not be undershadowed.
Good perspective.
And all this art is rooted in amazing science and technological feats. So I think it was like last week, NASA just announced that they are they paid, got 3 different service providers that will be sending things to the moon in 4 years.
That's right.
SpaceX is one of them that's going to be landing there. And with that contract, they're also going to demonstrate in-orbit refueling, which will enable them to demonstrate that their technologies can go further. Blue Origin is teamed up with the United Space Launch Alliance, and they're using the old proven Apollo kind of a multi-stage landing program. And then there's this third company that I'd never heard of before.
I've also never heard of them. Do they start with a D or something like that? Like,
right. Where did they come from? What?
I had never heard of them before.
But it's a cool idea how they say the astronaut can just step out as opposed to having to like come down from a massive rocket. And anyway. So this is all rooted in real science. And so the arts and the science bounce back and forth. They're not like mutually exclusive. You don't have to be in one camp or the other. And it's a shame that the historical academic system often makes people believe in that dichotomy.
And there you go. And not to take us too far afield, but in answer to your open-ended question on your Facebook, which rocket would I want to be on, or which ride? It's any of them. And the 16-kilogram requirement is an actual requirement. I want to talk to you more about that project. The Chinese try to grow something on the surface of the moon. I'd like for us to give it a shot. That's later in the decade here, but why not put that out? Let's learn more about the roots.
And so today, nod factors. And so I had to think, what is important about nod factors and legumes? Nod factors are chemicals that are found in legume roots, which allow them to form nodules. So what's the big deal about nodules? These things are amazing because they literally have the power of Thor inside them. What do I mean by that? Well, in order to get nitrogen out of the atmosphere is the N2 form, you need a lot of energy in order to create a bioactive form of the chemical. And it's essential as a building block for life, all life. And so lining is one of the ways that this occurs. Lightning strikes, take nitrogen from the air, and they create nitrates, which can then be absorbed by plants and animals. But this doesn't happen that often. And so humans have created a synthetic way of doing this called the Haber process. And it uses the same amount of electricity. So it's really energy laborious. And so we spend millions of dollars every year all around the world doing the Haber process, paying electricity to do it, and then transporting those nitrogen products to the fields all over the world. This is the modern intensive agrichemical, conventional industrial agronomy complex, which is good. We can eat cheap food, fantastic, I love it. But one of the things that we've learned is historically for hundreds of or tens of thousands of years, agriculture has existed without these inputs. And how did agriculture evolve to do that? Well, it was cultural practices. It was the changes in the types of seeds that we used. I once met this guy called Turnip Townsend. He was the son of Lord Townsend, the famous Turnip Townsend, the guy that invented the turnip rotation system, the 4-course rotation. It was the first ever 4-course rotation system written on paper in the UK. So he was very chuffed with his family's ancestry. And I went on and on talking about turnips and crop rotation. But having said that, it was right. Like the concept of changing the type of crop you have in the field every year and ensuring that it ensures the soil remains fertile. And what they didn't realize back then is they were like selecting for different types of populations in their environment, in their soil environment. And so as time went on, I wanted to learn more about how we could maintain soil fertility. And so I became interested in soil structure and soil chemistry. And this one little thing you see at the bottom here, this biochemical reaction here, is that Haber process, that fixing of nitrogen from the air. And these amazing genes, these nitrogenase genes, can basically do what Thor does with his lightning strikes. So understanding understanding that better is going to be really important for us ensuring agriculture remains sustainable in the future. And so we can, so we're going to learn a little bit about the process in order to know where we're going. Okay, cool. So at the moment, over the last few years, this is a graph. Graphs are very important to me as a scientist, but it's not too scary. We have time on the bottom axis, on the X, and on the Y axis on the left, we have the population of the planet. And on the right, we have the amount of fertilizers that we use. So you can see they're more or less directly proportional. And so as the human populations expands into the future, we're going to need to continue putting fertilizer in the field unless we come up with other ways of providing nitrogen to the plants. So from like 2020 onwards, this is modeled data, this is predictions. So humanity has the capacity to change these predictions if we come up with collective ideas that are good and worth implementing. Okay, cool. And so let's look at how naturally plants have evolved to work with their local microorganisms in order to help fix nitrogen from the air around them. So as always, my favorite hero isn't Groot. No. My favorite superhero is Arabidopsis, because if you look at its root physiology, you can learn some generic rules about all plants on this planet. And by looking really closely, we might be able to see some things that we can measure. So because as a scientist, I'm always looking for stuff that I can measure, because I don't always just trust my own judgment. I like to produce data to support these things that I believe. And so one of the ways we can create data is by looking at things really close. So a tool that scientists often have at their disposal is a microscope. And there in the middle of the screen, you can see we have a root looked at using a microscope. Isn't it awesome? It's growing there on agar, so it's nice and clear, and it's happy. If you get to go to college, you can sometimes play with really cool microscopes. There's one called a scanning laser confocal microscope, in which you can shine laser beams through the plant. These plants here that we're imaging, we've genetically modified them to put green fluorescent proteins in the cells that become root hairs, so we can see that specific cell type. So these confocal microscopes allow us to do laser cross sections, so we can keep the plant alive and growing and look inside it and see what its cells are doing. It's a really cool piece of technology. And that's why you should practice microscopy, or you should try at school, and because the more you practice it, the better you get, and the more crazy microscopes you get to use in the future. So we were looking at root hairs. What were we looking at? I forgot. Yeah, we were looking at root hairs there because they're really cool, particularly when they glow. But we can also look at the root tip. So if we want to know how big plants are, we can measure, say, the length of the entire root or the length of the entire shoot. But the thing that controls the size of the root is two main features. One, the meristematic region, the size of the apical meristem, and two, the final cell size, which occurs after the elongation zone, in the differentiation zone, when the root hairs have differentiated and appeared. So by comparing these two features, we can determine why the plant's roots are long, short, why they twist off to the left or to the right, or some of these other things that we alluded to in previous sessions. And my favorite part of the root is this, the quiescent center. So this is like the brain of the institute. Right here, there's there's a hormonal orchestra that's playing a symphony, where we have this auxin chemical, which is determining which way the plant will grow and how fast it will grow. But it's conducting with all the other hormones that are coordinating in this area to control the plant's growth and development, while as it takes all these inputs from these different sources. So don't get me started on this area. It's fascinating. So what happens when you put all that together? So this is a beam time-lapse photography, and there's something that's missing from this. You've got to watch this and tell me what's missing. Oh, Simba. Hey, everybody. So, okay, a few days in now. So this is from G-Phase. I must say, shout out to the photographers that took this image series. They have a fantastic YouTube channel in which they have time-lapse of many interesting plant thingies, and all done with this format. Isn't it great?
I guess, where's the water?
Just enough moisture in there. So there's probably some kind of wicking surface in the back that's bringing the water up from a reservoir below, a bit like hydroponics, I assume. They have, as an artistic YouTube channel, they haven't really written detailed protocols as to how they did it. But I think I showed a similar one to this earlier with because I was inspired by this and I tried to do it with cotton. Got that to work. It's not quite as good as theirs, but all part of the game. So this goes on for a while. So what is it missing? Okay, well it's moving around. That's cool. So that's you searching for light and things to hold on to. It's got a funny name. It's a chiral motion. I don't know. My mum would tell me, oh no, you know. So now looking closer, what's missing? No one's said it yet. What are we looking for? What's missing?
The nodulation.
There we go. There's no nodulation. Okay, so n equals 1, but what do you reckon? Why are we not seeing nodulation?
There's no bacteria.
Because the soil is enriched with some sort of nitrogen substitute or nitrogen in it.
Probably, right? Yeah, it's probably got lots of nitrogen in the soil, and they probably didn't add any extra inoculant, so it's relying on the background microbiota, like as the inoculation. So those two things combined, I mean, it's a beautiful time lapse, but we didn't get to see any nodulation. But there's some other things we didn't notice, and this is a wonderful thing. What they've gone and done is gone in again, and they've highlighted 2 different areas there, so you look a bit closer at them, and you can start to see things you didn't notice the first time. I just love these lateral roots as they come out. But there's a moment where you can actually see like the degradation of the bean. So you see what I mean? How like you can see the same video edited in a slightly different way and start to observe different features. So initially I was talking about just measuring like root length or like root hairs, but like there's all different places on an organism at different stages of development, and all of them need to be examined really closely because they're all fascinating in different ways. Look at that, the ways. And normally we're just looking at things like grow, but here we're looking at something like just degrade. Isn't that amazing? I guess, were they microbes they were eating? I don't know, but it's cool.
What is that?
Well, it was probably some of these microbes that were in the environment. Mm-hmm. So when you asked me to talk a little bit about nodulation, I was like, well, that's not really my area of research, but like education never stops. We're always here to learn. And I did study it in college. So I went, right, let's go back and visit the area and come up from my new perspective. My new perspective being a genetic engineer and a computer science kind of dude. So what I did was I found the most up-to-date paper in the area, and oh my god, it looks complicated. Loads of long words I've never seen before. I don't know how to pronounce. Okay, but that's okay. Don't worry, don't freak out. This happens all the time. Whenever you start a new project, you go to the literature, and if you've never worked in the area before, you're going to encounter some words that you haven't before. Don't be put off by it. This is the journey of life. Okay, but what we can do is find a few things to take home. So what I would do in the paper, I would take the abstract like this, and I try and reduce it to like 4 main bullet points, 4 take-home messages. This was selected by me because it says transcripts and genome in it, because I like genetics, I like genomes, and so I know I'm going to be able to apply my skills there, and I know this is like a modern paper, and the genomes, they have like a blueprint for every organism, how it develops and how it grows, right? So in that genome, there may also be a blueprint that determines how that organism interacts with other organisms, such as beneficial microbes. So that's why I wanted to have that component in the study. And then they say that they compile symbiosis-related transcriptomes. So the transcriptome is how the genome is being expressed. And so then we'll be able to see which parts of the genome are important to the formation of this symbiotic relationship. And that might tell us some important things about this symbiosis. In this study, they use 5 rhizobial species and 5 different like beans. So it's looking at the broad diversity. There's many different types of beans and many different types of microbes. So I like this because it was a meta-analysis. I was looking over more than one. And then finally, just a little bit of biochemistry, plant phenolic compounds that are actively synthesized and exported in response to nod factors. Okay, cool. So I'm gonna have to learn some biochemical names. So biochemical names. Chemicals are made by enzymes that catalyze reactions. We saw that earlier, right? When we were looking at the nitrogen fixation, that nitrogenase enzyme that has the power of Thor, right? Mm-hmm. So we might need to just get a little bit of knowledge surrounding that bit, a little bit of biochemistry and how these enzymes might catalyze those reactions. That will probably become clear in some of these pictures later. So that's my goal. I really wanted to understand that because that seemed interesting. So, okay, so as a plant person and as a computer person, I'm going to be breaking this whole thing down into sequences of little programs like that are being executed by the organisms inside. I like to compartmentalize things and simplify things, so this helps me understand things. So genetic program number 1, which is the plants will make flavonoids. plant roots, they exude chemicals. Sometimes they do this to make stones in soil, like release nutrients. And sometimes they do it to communicate with the microbes that are around them. So one of the root exudates is these things called flavonoids, and they can then interact with the microbes, which then cause the production of nod factors. Nod factors are chemicals that are produced by the rhizobia bacteria, which can then be perceived by the plants. So this is a molecular dialogue. It's a conversation that's happening in the water, with chemicals diffusing between these different evolved organisms.
Is this like an old school modem? Handshake?
Yeah, making those noises. Okay, cool. So we've got these nod factors. Okay, and these nod factors are amazing. They go over into the roots. They interact with the root hairs, and then there's a signaling perception thing that occurs in the hairs that switches on a bunch of genes that produces chemicals. So there's this positive feedback loop, as we call it. One good thing leads to another, and it happens again and again, and then eventually it sends a signal. And this was the thing I found amazing and really cool. It sends a signal to the shoots. So we've got all this molecular biology, and don't worry about the model down here too, okay? But we've got all this molecular biology going on in the cell. These chemicals are moving back and forth between the organisms. And then the plant sends a signal up to its shoots, up elsewhere in the plant, where that signal is perceived, another signal comes back down again. So why? What's that signal all about? Why does a signal go from the roots to the shoots and back again? Well, the roots are asking permission. They're saying, hey, leaves, we've got some bacteria down here who are hungry for some sugar. And can we give it to them? Because and then the shoots, if they're happy and they're growing and they've got like excess sugar, they're like, yeah, sure, we're producing loads of that stuff by photosynthesis. But you know what we need? We need some nitrogen. So make sure if you're going to give them some of our sugar, they're going to give us some of that nitrogen that they can activate. And so that's how the plants choose to have the right relationship and be mutually beneficial for the two of them. So that's why there has to be a signal that goes up and then comes down again. So not all plants have this program, okay? It's like not all computers have the same software. But this software does exist in legumes. And this is why legumes are such amazing plants. And so—
Richard, it's Michael. I just texted you a photograph that I literally just took of a 20-day-old Adzuki, Vigna angularis, of a whole mass of nodules. If you want to throw that up in the during your presentation, I'm trying to set up to do some microscopy with them now, but
Azuki!
that's enough, Gil.
I got that's 22, bro. That's 2 for 2. Okay, Richard, back to you. Just Azuki's winning. But, okay.
And now back to our regularly scheduled program.
But this is a really good point, though. So this is like the bridge between theoretical modeling and using science and experiments to understand the environment, understand what's happening inside the plants. But then this all has to be backed up with scientific methodology and scientific practice. And in order to continue to understand this, it's these exact skills of growing these legumes and quantifying their nodules, which people need to learn so that we can understand them better and use them more in the future. So there's another piece of software that's running back and forth. Okay, cool. So the roots have got the signal from the microbes. They've sent a signal from the shoots to the shoots. The signal has come back and they're like, yeah, go for it. And so they start to initiate nodule formation. The root hairs, they have these infection threads that start to grow through them as the bacteria start to occupy the cells. And this is the bit that I really do love. Okay, so what's happening there as the rhizobia is entering the root hairs? We're seeing what we call calcium spikes. Calcium spikes are electrical oscillations that are moving along the membrane of the cell, which are causing calcium to move in and out of the cytosol. So this is your Morse code that's going through your plant. This is its nervous system. It's a shame the neuroscientist isn't here anymore. I should love this bit. Okay, cool. So what do we have? So there's always natural oscillations of electricity going through the plants, but are never intense and periodic like this. So what we see in the top right-hand corner here, we have a bunch of, this is time-lapse photography. The 5 seconds, 10 seconds, 15 seconds. So we have these different frames going through as we've imaged this one root hair cell on the surface of the plant. It's being infected by these by these rhizobial bacteria, and it's starting to recognize them. And we know that because of this peak, and this peak, and this peak. And it comes at this regular interval. You can also visualize that not in like a spatial form, like this, as the hair. See, this is the hair, the spatial information. But you can now just imagine it as an intensity of the electrical wave or the calcium wave. You can see those peaks. So this is what we call calcium spiking, also known as nuclear spiking, because this is happening in the nucleus. You can see it by the distribution. Root hair cells are long and kind of like stretchy things, but you can see there's a peak in the center, and this is where the nucleus is. The nucleus is where DNA is being actively transcribed. So there's something about the interaction of the microbe and the plant that causes this electrical oscillation along the cell, which causes mass amounts of calcium to change gene expression patterns in the nucleus. And this then causes the next stage of the process. So I took that root hair coiling and projected the rest of the model. So then you get the initiation. The initiation is the change of the elongation pattern within the root hair cell, and essentially the formation of a nodule. In order to do this, there's actually some hormonal regulation that occurs. The microbes actually change the hormones inside the plants in order to ensure this happens. Ethylene is a really interesting hormone. It's a critical signal in the response to flooding. So that's going to be important a bit later. And ethylene regulates this whole process. So if you have high ethylene, you can prevent this process from occurring. Anyhow, so, and then eventually a mature nodule will form. And we want that mature nodule because the mature nodule, well, that's where the magic happens. So this is where the next software program gets activated. And this is nodule fixation. So this is a different genetic operon, a different set of genes that get turned on to produce a different set of enzymes, which then actually start taking nitrogen out of the air and fixing it. FYI, I can't see the comments, so if anyone has a comment, and feel free to shout out. But this is the exciting moment. Okay, we've now gone through all these different genetic programs, and now we've got a nodule that's formed and is able to fix nitrogen. And don't get me started on those genetic operons, because these hormones here, like gibberellins and auxins, this is what my dissertation, my thesis was on. So there's so many different detailed molecular tangents that you can go off on. But the moral of the story is this was that hormonal change I was talking about, and this is what's causing the tumor to form. So essentially, the bacteria is activating the formation of a tumor on the side of the plant, which they will then occupy. This is not only done by rhizobia varieties in order to fix nitrogen. This is also done by Agrobacterium tumefacium, which is the model microbe that humans use to genetically modify plants. Okay, cool. So we've gone through a lot, haven't we? Oh, got it. Okay, let's have a quick summary again. Plant root hairs produce flavonoids, get perceived by rhizobia, that build nod factors, that communicate with the plant, that cause a nodule to form that allows them to fix nitrogen. That's on the syllabus, I'm sure. So again, just to pause and reflect about this, there's a lot going on, and all I've been showing you so far is pictures. The reality of it is a little bit more chaotic, but it's basically the same. You'll see they're not always quite so beautiful, and getting the right stains at the right time is a real art form. Yeah, this is a scientific method published in Nature Reviews, but I'm calling it a real art form. What they're doing here is they're staining with a GUS gene so that they can see the infection of the fungal hyphae. In order to get that stain just right, it takes practice. So you have to give it a go. You have to be able to do it wrong a few times. And so the net scientific process actually has a lot of art in the basis of it. So pause to reflection on the science that we've gone through is basically to inform you as to why it's really valuable for you to understand these methods that you're being taught through the ExoLab program. Okay, cool. So, but our nodules, oh God, nodules are amazing. But what makes the nodules so special, eh? Well, inside the center of the nodule, you get an anaerobic environment that forms. Where you've got air, you get gaseous movement, you get convection, oxygen moves around, and all these other things. In the center of a nodule, you don't get that. You're surrounded by cells and surrounded by this dense area of water. So actually, right in the center, you get an area of anaerobic anaerobia that forms, a local area of hypoxia that forms. The cells start to experience a little bit of anoxia. They start to suffocate a bit. There's many ways to say that oxygen is depleted in the center. And the reason that's important, it takes us back to biochemistry again. So this nitrogenase enzyme, remember the enzyme that has the power of Thor? can fix nitrogen. Well, it has a weakness. All superheroes, like Superman has his kryptonite. Well, nitrogenase has its oxygen. And oxygen can competitively inhibit it by getting into the active site where nitrogen normally functions. And so it prevents it from functioning. So to get a functional nitrogenase reaction catalyzed, you have to reduce the amount of oxygen in the area. So this is like the three-dimensional structure. People have been looking at this for a long time. Three-dimensional structure, that nitrogenase enzyme, and buried away in the middle here somewhere, there's a spot where nitrogen goes in and then like nitrites come out. And if oxygen gets in that space, it blocks it. So the higher the oxygen you have, the less nitrogen fixing you have. There's a schematic to kind of represent that. Okay, cool. So why is that the case? That's weird. Something as important as nitrogen fixing from the atmosphere, surely you'd think evolution would come up with a better mechanism, something, because oxygen's in the air all around us. So it'd be crazy to evolve an enzyme which has a constraint such as oxygen in the environment preventing it from function, right? That would be crazy. So this is when we go back in time. You know, my science talks, they always take us back in time, sorry. So we're going to go back like 2,500 million years ago, back to when like the first like bacteria were like just first fixing like carbon dioxide from the air, making their first like sugars. And in this early period, these early bacteria, these cyanobacteria, these archaeans, they had low oxygen. Can you see that on the right here? So this graph has the atmospheric oxygen concentration in it. And so we have our like modern time up here kind of thing, but you can see all the way back there at the beginning of this cellular evolution, the amount of atmospheric oxygen was really low. And the need for these cells to grow, the one building block they really needed was nitrogen. So the selection pressure was such that they evolved the capacity to fix nitrogen, and they didn't have oxygen accounting against it. Time then passed, oxygen levels changed, the world evolved, but these enzymes, they just remained as they are. There's a similar story regarding Rubisco and photosynthesis, but that's a different tangent. So evolution is everywhere. Okay, so the moral of the story though is oxygen inhibits nitrogenase, and oxygen is the kryptonite to Thor's lightning. So where naturally on the Earth does low oxygen occur? Maybe there is another opportunity for this type of process to evolve. You have convergent evolution, you have like where things come together. Something is so important, you can come up with the same system using slightly different mechanisms. If you know what the core fundamental principle is. And here, it's oxygen availability. There's a clue on the left here. You see these bubbles of water? Or this plant that's thirsty, but not that thirsty. When you submerge a plant, it goes into a low oxygen environment. So let's start thinking about like moisture as being a temporary way to create a hypoxic environment that could allow nitrogenase to function. And at this point, I'm gonna watch a video because it's awesome. Right, cool, huh? So
Absolutely amazing.
Okay, so Fenway, Jean-Michel Anne and his team and his collaborators have gone out there and they've looked at natural diversity of corn, and they have found a variety of corn that naturally form symbiotic relationships in order to fix nitrogen from the air. It's awesome. Okay, so the majority of the food produced on Earth that feeds the metropolises that most of humanity live in, it comes from monocots. Comes from grasses like corn and wheat and maize, barley. Oh, my favorite. Oh, sorry, my favorite beverage. All come from these monocots. And we spray a lot of nitrogen on the fields every year in order to ensure we have lots of them, so that our favorite foods are cheap. That's great. And the problem is, and we do crop rotation in these fields. These farmers are doing their best, and soybean has been selectively bred to become a very efficient crop. But perhaps we could find ways to put less artificial inputs and agrochemicals in the agro ecosystem. And maybe by enhancing certain crop varieties to incorporate these natural symbiotic relationships and enhance that, we again might be able to reduce artificial inputs into our agro ecosystem, thus making them more sustainable going forwards. Okay, so we have this natural variety of corn. I've seen these plants growing in the wild in a greenhouse in Madison. These things, they're like 2-3 stories tall, and they had a tiny little pot like this. It was awesome. You have to spray them with water. Anyway, the moral of that story is they are distinctly different from the elite corn varieties that we have in the fields, well, being sown probably right now in a field near you, right? So there's going to be a lot of work that's needed to transfer this trait over, and to do so in a way which is productive and better for the environment than the current status quo. How are we going to do that? Because currently we have fields that we're feeding. You know, in my opinion, corn is the dominant species on the planet, because humans have spent so much time and energy combating its natural competitors and selecting for it. And we're doing really amazing things to increase the yield on the land that's already under cultivation. Because of these projected increases in human population size, we need to make the land more productive. So you can see the distance between these plants here, and then here, and then here. I know one professor who has been making the plants colorblind, so they can't perceive the shade of their neighbors. So you can sow the seeds at twice the density. I'm getting twice the yield from the field. It works, but also pushes more water from the ground up into the air, which might be the reason we're getting massive rainstorms in the Midwest at the end of the summer each year, because we're downstream of the Corn Belt. But we'll just have to see how many years that pattern continues for before it becomes a scientific belief as opposed to a weather-based phenomenon. It's been 4 years running so far anyway. So there may be other ways that we can apply the use of genetic engineering technology, which rather than making the fields require more resources, instead actually makes them require less input. And one of the ways I believe we can do that is by thinking about the field as an agro ecosystem and try and enhance the diversity of that. And so perhaps we can create new varieties of microorganisms and new varieties of crops to communicate with each other better.